Anyway, thank you very much for the introduction. I want to also thank all the organizers, the staff, and the volunteers, and the tremendous effort to put this MACNA conference together. It's just been great. There's so many. It's been fun for me because there's so many colleagues and friends and kindred spirits. I'm not sure what a kindred spirit is, but I'm sure that's all the rest of you out there. But it's great to be here tonight. Well, listen, when John Coppolino called and asked me to be the speaker tonight, at first I said, well, you know, I can talk about the politics and regulations of fish collecting in Hawaii. It's really a big issue right now. Silent pause. And John was very polite, weren't you? You said, maybe not. I kind of want the audience awake, you know? So, okay. He said, maybe if I have some photos and videos, things I've accumulated over the years, maybe I could entertain you a little bit with that. So that's what I'm going to try to do and hopefully keep you awake too. As you have already heard, I have retired from my professional career, but I still do much the same kind of work with fish and everything. I just don't get paid for it anymore. And the title of my talk tonight is Some Adventures, Discoveries, and Mishaps in the Career of an Aquarist. So with that, I'll just go ahead and get started. My passion for aquariums as a career grew from my desire to share with others some of the amazing things that I've had a chance in my career to experience underwater. I could have relied on photos and videos and maybe movies and things to do the same thing, but I chose aquariums as the vehicle to help people to understand and hopefully to care for the life of the oceans. Most people will never put on a mask and snorkel and go under the ocean, so aquariums are really our way to bring the ocean to everybody else. Anyway, my professional career, when I actually started getting paid to do this work, was 1976 at the Waikiki Aquarium. I was there for 26 years, and more recently at the Georgia Aquarium in Atlanta. I was there until 2011, and then I retired and moved back to Hawaii. Over the years, I really developed a number of rules, guidelines, whatever you want to call them, ethics even, in developing exhibits. And I'll share these with you. It's really the only text slide that I have. But I always felt that exhibits should provide, first of all, the best possible environment for the animals. That's number one. But also they should exceed visitors' expectations. And I'm basically talking about public aquariums, but this could apply to a home aquarium as well. That they should make a positive, emotional connection with visitors. And that they should inspire and challenge visitors to engage in conservation. And they should encourage us, as professional aquarists and even hobbyists, to give back to nature. Actually, a lot of the stuff we put in our aquariums comes from nature. We need to give back to nature, supporting and conducting our own conservation programs and research. Whenever possible, at least at Georgia Aquarium when I was there, we tried to use captive bred, rescued, and rehabilitated animals. And lastly, to never misrepresent the true nature of animals. And by that I mean, Finding Nemo was a wonderful movie, but it's really not the true nature of animals. And that's sort of what I'm getting at there. In a public aquarium, I think we have an obligation to represent the true nature of animals. So what I'm going to do now is kind of go backwards in time, starting from my most recent job and working back to the very beginning, and then I'm going to move forward again. So here's kind of a quick evolution or de-evolution of aquarium exhibits. This is the last big exhibit I designed. It's at the Pacific Barrier Reef at the Georgia Aquarium. It was 164,000 gallons. At the time, it was the largest living reef exhibit in the United States, maybe the world. And Bart Shepard at Steinhardt said, when ours is open, it's going to be 164,005 gallons, just to make sure it was biggest. And we used, keeping with this, using cultured animals and so forth, we used cultured reef rocks. 5 metric tons that we got from Walt Smith. I ordered them a year or two in advance, put out in the ocean. 5 metric tons of cultured live rocks. And nearly all the corals on opening day were cultured or they were obtained as confiscations from the U.S. Fish and Wildlife Service. There's one Rumpfella gorgonian in here, Sonia. If you're around here, you'll appreciate. In that little circle, there is a Rumpfella gorgonian. It's significant because I collected it in 1982. And the parent colony's long dead, but this one I've kept with me most of my career, actually. Now I'm going back to Hawaii, going back in time, 2002. Before leaving the Waikiki Aquarium, Charles Delbeck and I created this living reef exhibit. And I really have to credit Charles and Rick Klobuchar for fulfilling the goal we had of creating this living exhibit, since they were really the caretakers after I left. But the giant clam in the foreground is one I obtained in Palau in 1982. It was 5 years old then, and it's still alive today. It's 38 years old, and it's the oldest and largest giant clam in any aquarium in the world. Going back further in time, 1996, I set up this outdoor exhibit to feature corals at the Waikiki Aquarium. We were culturing them by that time and distributing them to aquariums all over the place. So we called it a coral farm. Actually started out with a little egg crate and frags and so forth, but it really got out of control. This exhibit is still in existence at the aquarium. And that imperial angelfish was given to me decades ago when it was a juvenile. There's one person in this audience who would probably know the person who gave it to me. Gustav, if you're here, you might know Kuni Sakamoto. He was the manager of the lab at the University of Guam. I was running through Guam, and he said, I want you to have this little imperial angelfish that I collected. So he gave it to me. This is early 1980s, and that fish is still alive today. And that rumfella, that's the same one from 1982. We set up this intermediate exhibit in 1992 just to be sure that we could manage these corals if we ever built that $100,000 exhibit later on, which we did, and the rumfella again in the middle. Going back to 1990, by that time we had learned to keep Acropora, and we had to set up more and more of these outdoor tanks. They were just, as you know, they grow very fast. We didn't have any place to put them, so we just kept adding more and more of these tanks. I'm really happy to see today that there are some coral farms today use the same model you see here, but on an industrial scale. So this kind of system is still in existence. Now we're going way back. This is 1980. At that time, we had really mastered large polyp stony corals, but we really hadn't developed current and surge devices that we really needed for the acroporids. They're still beyond our ability back then, except for one acroporid, Acropora brueggemanni. I knew in Palau, in the backwater lagoons where it's very calm, there's really not much water motion. There was one acropora that lived there. So I collected that one, as well as these goniopora, and they're still alive today at the Waikiki Aquarium. Anyway, these corals, these three colonies here, I think are the oldest Pacific corals in any aquarium in the United States. If somebody has something older from before 1978, let me know. Oh, and also, I don't think any aquarium had ever displayed a cuttlefish in the United States prior to 1980 either, so that's probably the first. Way back, 1978. And this is the first Pacific Reef exhibit, I think, in the United States. I set it up at Waikiki Aquarium, and it was based on a tank I'd set up even earlier in 1972 in Fiji. Back in 72, Peter Wilkins was developing closed system aquariums in Europe. I don't know who in the United States was working on it. I was inspired by Li Chen-Eng, who had just about that time published his papers. And I'm going to go back a little farther, and this is what I was doing. This is 1964. Nine months of winter in Michigan for 20 years and shoveling snow. It probably had a lot to do with me moving to the tropics. But the tropics and coral reefs was really my destination, because while I lived in Michigan, I had my saltwater aquariums. And this is where it really all started for me at age 13, 1962, when I set up my first saltwater aquarium. Not this particular one, this was one a little bit later. But they were really my inspiration to someday become a marine biologist. Growing up in Michigan, snowy climates, and I'm sure there's a lot of people here the same thing, especially kids, I hope, that these aquariums, you start dreaming of exotic places, hoping someday you might get there. And like Charlie Barone said yesterday, we know many other professionals who got their career working with aquariums as a child. So this is where I started 53 years ago with saltwater aquariums and freshwater aquariums before that, of course. But now I'm going to start moving the clock forward again and just tell you some of the experiences I had after I quit shoveling snow in Michigan. I joined the Peace Corps. After graduating from the University of Michigan, I was assigned a project in the Fiji Islands, and I had to look up on a map and find out where Fiji was. I moved there in January 1972. So this is the Fiji archipelago, and believe it or not, Walt Smith and Tony Nahakiai are not employees of the Fiji Visitors Bureau. Just by coincidence, we all ended up in Fiji and got to know each other there. So Fiji archipelago, it's comprised of 337 islands. I got that number from Walt earlier today. And I was stationed here in the capital city of Suva. It's coming. There it is. And I worked at the University of the South Pacific. We called it USP. And I want you to keep that acronym USP in mind. You know why it's probably going to come up again, of course. But keep USP in mind. Well, my assignment while I was there was to conduct a survey of the marine fauna and flora of the archipelago and to set up a reference collection for the university for research and teaching. And there it is. When I arrived at USP, the Marine Survey was already well underway. Peter Beveridge, New Zealander, was in charge, and fellow Peace Corps volunteers Rand Dibdahl, Mike Gowell, and Lance Craig had already done quite a bit of work. My 4 years, we surveyed fishes, corals, mollusks, echinoderms, arthropods, and algae. The collections back then were kept in 2 buildings left over from World War II. But I'm really happy to know here it is 40 years later, and the collection still exists. The university's built a climate-controlled room in their new marine sciences building for the collection, so it's been quite valuable. Now I want to show you some of the little fish discoveries we made back in those days. I'm going to start the video. Hopefully it'll play. I can't really see it. Well, I sort of can. Yeah, I can see it. I found this. I love blennies, and I got to love it partly because of this fish. I sent it to Vic Springer at the Smithsonian, or maybe it's over there. I don't know where I am. And eventually he decided, oh, this is a new species. He called it Exenius fijiensis and described it in 1988. Great little fish, and Walt, you ought to export these for people who are nano reef aquariums. It's endemic to Fiji. Aha! So you get the market cornered. And now this is why you have to remember that acronym USP. When I got there, Mike Gowell and Dave Woodland had already described Siganus uspi, and many of you may have this fish in your aquarium, thanks to the folks in Fiji who send them out. USP, University of the South Pacific with the I. So that's how that got that unusual name. The first fish I actually discovered, and you may not be able to actually see it, I was working on crinoids, sending them off to get them identified. That crinoid is Comanthus benneti, and right in the center is a little fish. And it popped out of the crinoid in the bucket. Well, that's unusual. Turned out it's a clingfish, so I sent it to the expert on clingfishes, Jack Briggs. In 1976, he described it as a new genus in species, Discotrema crinophyllum. Not one you're likely to keep in the aquarium, but everybody who's a diver gets to see these. And one of the last fish we discovered was this Goby valenciana decora that Doug Hosey and Helen Larson described in 1994. Now, moving on, in January 1974, Dr. Barry Goldman, he arrived at USP, and Barry had just described a fish that I thought I was discovering brand new, and then I did the same year, but Barry already described it, the magenta dottyback, Pictochromis porphyri, which is quite common in Fiji. Roger Lubbock, the late Roger Lubbock, and Barry Goldman described it in 1974. Anyway, Barry and I became good friends, and Barry arrived in Fiji from Australia by sailing there on his yacht, the Bundoon. Now, some of you already know where the story is going, but for those of you who don't, now I'm showing you pictures to go with it. Anyway, in July 1974, Barry said, hey, let's go out to the Lau group in the southeast section of Fiji, which in the 70s really had barely been touched and explored for scientific purposes. And so we planned a trip. Barry Russell, a fellow Aussie, and Lois Goldman joined us on the trip. This was our intended destination from Suva to the Lau group. If you extend that arrow a little farther to the southeast, you end up in Tonga. And this is the route Barry plotted to go just north of Moala Island, and this is the route we actually took. Paying attention, that line suddenly stops. Right in the middle of the night. There we go. Right in the middle of the night, we slammed head on into Navatu Reef. We were sailing at 9 knots. It was strong wind, good clouds. There was no moonlight or anything. It was pitch black. Actually, I was at the tiller. And Barry said, just keep on this compass heading. And it was just that little green light with the compass, with the tiller and everything. And suddenly there was a white line in front of the boat. Could just barely see it. And I knew immediately, just as soon as we hit it, it was the reef. We slammed right into it, 9 knots. Every wave would come, would pick the boat up, crash us onto the reef crest. Then the wave would recede, the boat would flop over on its side. We were all hanging onto the deck. And then the next wave would pick us back up again, slam us down on the keel. The boat would flop over. This went on for hours until sunrise. And if that boat had been made of fiberglass, or if it had been made of wood, you would have had to have a different speaker this evening because I wouldn't be here. But the Bundoon is a steel-hulled boat. Thank goodness. Anyway, this is what it looked like in the morning at sunrise. But by a miracle, because most boats, when they end up on the top of a reef, they stay there. At high tide, the boat righted itself in a little sand channel. You can see it. I took a picture of it. And we had just inches to spare under the keel. It was that much of a margin, or little margin. The surf doesn't look very big in the background there, but it's probably because of the distance. But it was really slamming us on the reef. The other thing there was no land. If the boat had crashed, there was no land to go, wait for a rescue. That would have been it. And Barry said emphatically there was only room on that dinghy for he and Lois. So anyway, we got into the lagoon, and the other thing, once you're in the lagoon, is there a way out, But fortunately, the trade winds come from the southeast, so in the northwest corner there was a channel to get out. That's where you'd expect to find it. Well, we decided to make our way back to Moala. There's safe harbor and a village there. And these are my field notes from July 2nd and 3rd, 1974. And we collected, of course we went fish collecting, even, had this terrible accident, but yeah, came out there to collect fish, so we're still going to collect fish. And we found this Miacanthus. On my notes, I said it was dark gray-green with a yellow stripe, and here's an underwater picture I took of it that day. Many of you may know what this fish is by now. I sent it to Bill Smith-Veniz, Pseudantheus Smith-Venizi, okay? Bill Smith-Veniz worked on fangtooth blennies, saber-tooth blennies, and I recommended to him that if this was a new species, you ought to name it in honor of the boat that didn't sink, because if that boat had broken, again, I wouldn't be here and we wouldn't have discovered the fish. So Bill named it Miacanthus bundoon, and if you have this fish in your aquarium, now you know how it has such an unusual name. And I see that ORA on their website has the ORA bundoon blenny from shipwreck to captive bred, so I guess that's a plug for ORA, but anyway, now you know the rest of the story. But that wasn't the only fish we found that day, and there's even a bigger mystery in this one, and this will take a few more minutes to go through. The other fish we found was Centropyge heraldi, and in parentheses I wrote with a black dorsal spot, but I'd already figured out it was heraldi, and this is that fish. Is it playing? Yeah, okay. So some of you, or many of you, have probably seen this fish with the heraldi, yellow with the black spot in your aquariums. But back in 1974, it was new. The first time I saw it, I thought, a new species. I get to describe a little Centropyge. This is great, Well, after we did catch him, I said, no, it's just heraldi, the all yellow one. But for some reason, the ones in Fiji have a black spot. 11 years later, the fish is becoming a common sight thanks to Tony Nahaki, who's collecting them regularly in Fiji and sending them back. And I figured I'd better write up an article about this fish and explain my conclusion that it's just a variant of Centropyge heraldi. I don't think you've got a new species. And this is the article I wrote in FAMA in April 1985. But sure enough, 1998, another, 11 years or so, someone eventually picked up the fish and decided it was new, and that was Rudy Cuter, and he described it as Centropyge woodhead eye in 1998, and he used the fish that we collected at Moala Island 24 years earlier as the holotype. He got it from the Australian Museum where Barry Russell had deposited it, and used that fish as the holotype. Rudy said he found differences in the head length and the fin length compared to typical heraldi, and the color pattern, and so he was convinced it was new. Well, Jack Randall and I weren't convinced, so we got back on it, asked Tony Nahaki to collect more of the woodhead eye, and we did a broader traditional analysis looking at both forms, and we couldn't substantiate any differences in fin shapes and measurements and lengths and so forth. So we sunk woodhead eye and said it's just a synonym of heraldi. And that was in 2000. Is that the end of the story? No. In 2009, this goes on and on. I mean, it's just one little fish, but Steinke et al., writing in PLOS One, the online science journal, reported on the basis of DNA barcoding. They were trying to identify tropical species with DNA, like fingerprints, barcoding a variety of tropical marine fishes, and they detected a DNA divergence between samples of heraldi and the woodhead eye from Fiji, and suggested or recommended that maybe woodhead might be valid. However, right now, one gene, a mitochondrial gene, is not sufficient evidence to say it's a distinct species, but it is suggestive that more work needs to be done. So the challenge remains to collect specimens of all forms of the species throughout its range and conduct both traditional and genetic analyses so we figure out what the heck's going on, because it gets weirder. Kevin Cohen allowed me to use this picture, and prior to that, Walt Smith had collected some weird fish, and I'll show you a picture of that in a minute. Kevin just sent me this a week or so ago, and he said, well, so what's this? This looks like heraldi, and it looks like the woodhead eye, but there's more black and woodlet, and the usual color on the anal fin. And I looked at it and thought, it looks an awful lot like it's got some bicolor in that anal fin. So look at the pattern on the anal fin of bicolor. You can see it just on the edge, it's the blue line, there's a little orange, and then it breaks up into spots. So fade back to that one. Anyway, that's not evidence of anything. It's suggestive that maybe there's some hybridization between bicolor and heraldi going on. And in fact, talking to Louise, he said those two species are, in fact, very closely related, bicolor and heraldi. And it gets more. Lemon sent me this giant heraldi, so they really get big for some reason, and he didn't know what it was, and there's more color in the anal fin on that. And Lemon even published this in Reef Builders in December 2014 with all these different color patterns. And Walt, there's your double black fin, giant heraldi, new species, hybrid, whatever it is. Anyway, so Lemon put this all together, and I want to quote Lemon in his own words, because he's made probably the most astute comment about these fish. Quote, "You'll want to break down and cry in confusion." Lemon, thank you. I couldn't have said it better. 41 years later, after a shipwreck, and we're still wondering what the heck that is. Well, anyway, for now, I'd say they're all variants and hybrids, and someday it'll all get figured out. And Charlie, as you know, evolution among reef animals is often a very messy process. There's evolution going on here. It's very inconvenient for us taxonomists, though, who want to know this and this are separate, but corals and fish, we see the same things. Okay, well, I didn't get a chance then to describe a new Centropyge. I had one other chance, and I blew it. In 2000, my wife, Marge, and I made a trip along the north coast of Viti Levu. All the years before, I'd been in the southern area, and here was where we ended up, in Bly Water. And yes, that's named for Lieutenant William Bly, captain of the Bounty. He was the first European to unintentionally navigate these waters after the mutiny on the Bounty, when he and 18 others were set adrift just off Tonga, down to the southeast. So he somehow or other navigated all the way through these islands. He missed Navatu Reef that we hit, but he didn't know the area. He didn't have any charts, and he got all the way through these, the first European to go through there. And he actually charted all the islands. I mean, Walt, you and I have talked about this many times, like how in the world did they do this? It's an amazing story. Don't have time to go into all of the details right now, but read about it. The account of the navigation through here is just an amazing story of survival and navigation. But anyway, I made my first trip there, and we stopped at Wakaya in this big reef system in that area, and found this little black angelfish, although it wasn't really black. Is it plain? Yes, it is. Okay. And I thought it was Centropyge nox, that's that black angelfish there. But when I looked closer, it was actually a deep blue color. I wrote to Jerry Allen after our trip. I said, I think this might be something new. And Jerry wrote back, said, no, he had seen it too. No, it's just Centropyge nox. And so I didn't have any specimens, and I just, okay, forget it. Anyway, a few years later, Walt started collecting them. Tony might have collected them earlier, but there's an odd thing that fish on the north coast of Viti Levu, for some reason, there are some different species than there are in the south. And that black form, I don't think, or rarely gets around to the south, so that's why we had never seen them before. I can't figure out why. Anyway, Walt sent them out, and in 2012, Kang Ning Shen et al. Described it as a new species based on morphological and DNA analysis, and Walt recommended they name it after Deb, and Deb Smith, and so now we all know this is Centropyge debra. Well, just a day later, another nearby reef, Marge and I found this new Cirrilabris. There it is. And it was also clearly something brand new. In 2003, Jack Randall, Jerry Allen, and I named it Cirrilabris Marjorie in honor of my wife, Marge. And I surprised her with this on her birthday. All right, last few minutes of my talk, I'm going to shift gears completely away from fish and even corals. So this may not have as much relevance to reef keeping, but it's an important animal that I want to talk about. It's been much in the news lately. There's concern about its survival in the wild, and this is the chambered nautilus. So let me talk about this for the last part of the talk. In 1976, just outside of Suva Harbor, I collected the first living nautilus I'd ever seen. I had always wondered where they lived. We lived there all those years, and we never saw a live one. Didn't know that they just lived really, really deep. And I was inspired by Doug Faulkner, who was writing in the January 1976 issue of National Geographic magazine how they were collecting them. I also knew the New Maya Aquarium, New Caledonia, had kept them alive in their aquariums for many years, but they really hadn't published anything about husbandry, so we didn't know if we could ship them in boxes or how to keep them or anything. This is the Fiji nautilus, Pompilius. And I brought our first nautilus to the Waikiki Aquarium, but I knew we had to find out more about the biology of nautilus in order to create a better environment for them in captivity. So it wasn't enough just to bring him in. We don't know much about the biology of this animal. We're going to have to go back in the field and conduct more research so we can make sure we create the best environment for the animal, my number one rule. When we set our traps, one of the easiest things we did was attach a max-min thermometer on the trap. So this will record the minimum temperature at the depth where the animals were collected. And it was cold. I was absolutely shocked how cold it was. I'll get to the temperature in a minute. But we also tracked animals with transmitters to see where they went. There was stories that they migrated, but nobody had actually tracked one to know for sure if they did or not. We demonstrated that they move from deep water around 1,000 feet during the daytime. They stay very deep. And around at nighttime, just after sunset, we track them with the transmitters coming up into shallow water in Palau at about 275 feet. In New Caledonia and some other areas, they come up much shallower at night, and that's because of temperature. It's colder in New Caledonia. In Palau, it can be 82 to 84 degrees in the surface. And nautilus don't like temperatures much above 75 degrees, so they don't come any shallower. And also, that migration from 1,000 feet, the water temperature can be 50 degrees Fahrenheit or colder. And within a couple of hours, they migrate up to 275 feet where the temperature is 75 degrees. So these animals can withstand tremendous temperature differences and pressure differences. Well, we published our results on the transmitter studies, and we also went back and started cycling the temperature in our exhibit. We couldn't do much about pressure, but temperature we decided to cycle from cold water to warm water and back every 12 hours to kind of simulate that migration they're used to. Now, the other thing is, even in 1976, as soon as we got them, they started laying eggs in the aquarium. But nobody had ever found a nautilus embryo. And even to this day, nobody's ever found a hatchling in nature, and nobody's ever seen an egg in nature. We finally figured out, and it took 15 years to figure out, we weren't giving them enough time to develop. We're used to cuttlefish and octopus hatching in like 30 days. Nautilus takes a year. A year. And this is the first embryo ever seen. The one in the upper part there looks like a wad of chewing gum. Just so you know what you're looking at, that is the embryo growing on top of a huge yolk. And then the picture in the lower right is a little nautilus. You can see its eye. You can actually see a siphon. Looks like a limpet. There might be something said for that. And of course, the egg capsule has been cut away so you can see it. Anyway, we published these observations too. There's a hatchling. They're miniature adults when they hatch out. They're the cutest little things. The study on the embryos went into Science, and then later Marge actually hatched the first one. She kept the incubator tanks, and we published that data, or those data too, back in 1980s. Now, fast forwarding to today, and the focus is now on the survival of nautilus. Hard to believe from the 70s when barely anybody knew about them, and today we're talking about their survival. Literally tens of thousands of nautilus are collected every year and killed for their shells, which are sold in shell shops around the world. So you can see those beautiful shells for sale, these. This is Honolulu just a week or two ago. $45, $46. Beautiful shells, but each of those animals was killed and tossed out for the shell. And I thank Greg Berard for that picture in the upper left, which was taken in the Philippines. Peter Ward, a colleague of mine who's been in the news a lot lately because of recent work he was doing with Alonautilus scrobiculatus. This is the fuzzy nautilus. It's the rarest of all the species of nautilus. It's so distinct it's in its own genus, Alonautilus. And so far it's only been found in Manus Island and Papua New Guinea, but I suspect it's also found in the Solomon Islands. Now, the reason Peter's been doing this work, he's been out in the West Pacific for several years sampling nautilus populations, gathering data on the number of animals caught per trap so that we get an idea of the populations around the Pacific. We know that in the Philippines, some populations have been totally exterminated. There are no more nautilus, even though we know earlier expeditions in the 60s and 70s, the Alpha Helix especially, that they were quite common. You go there now, you don't catch anything. These data will become part of an effort next year to consider nautilus for protection under the Convention on the International Trade in Endangered Species, or CITES. Marge and I have also been part of this effort, and this past May, we traveled to Palau to work on the endemic nautilus there, Nautilus bellauensis. This is Palau, and I put Fiji in there, and you can see Australia and Papua New Guinea, and Hawaii is way up on the top part of the globe. And here's the archipelago. We did all our trapping on this reef. Can you see the little yellow circle there? The Palauans know it as Mutrendu. I'm not sure that's pronounced correctly, but we all call it the short drop-off because it's just a short boat ride from town. Now, Palau's where we did our transmitter studies in the 1980s, and at the same time, our other colleague, Dr. Bruce Saunders from Bryn Mawr, had conducted an exhaustive multi-year study on nautilus in the same area. And his assistant's Mike Weekly, who was one of our employees at Waikiki Aquarium. But Bruce Saunders was primarily interested in growth rates over time, but he also accumulated data at the same time on catch per unit effort. And I saw him 1 year ago here in Washington. We had a conference on nautilus held by U.S. Fish and Wildlife Service over this whole issue, should it be protected. we said, you know, we could really go back and duplicate your studies and see if we get the same catch per unit effort, and that would give us at least an indication if that population has changed much or not. So that's what Marge and I set out to do. I contacted Neil Landman at the American Museum, and he gave us a small grant, and I contacted my good friend Pat Colin at the Coral Reef Research Foundation in Palau, and we put together this little expedition. When we arrived, this is actually a video, but I'll play it in a second. Pat is busy here working, finishing the trap. The traps were identical to the ones that Bruce Saunders used. We used the identical bait. We went to exactly the same sites to release the trap. In other words, we wanted to keep all the variables minimized. So if there were any differences in collection rate, it would be because there were differences in the population. Oh, and one other thing, it's Pat Colin and his wife Lori Colin, and many of you are familiar with his the fish named in his honor, Colin's angelfish, Centropyge colini, not colini. All right, let me run the video. So Pat's busy grinding the edges of the trap. I think I had a little picture of the angelfish up there. And here, Marge and I, Lori's on the left, and Marge and I are putting the finishing touches on the trap. This is the research vessel. And here we are heading out. Matt Wendell and his brother Zach are with us. I invited them out on the boat that day. And I'm baiting the trap, and we're heading out to the short drop-off, one of my favorite places to go diving. Marge is doing all the work. She's tying the trap line off while I take pictures of her. So, and here's Matt helping us out. And Sanji, this is not photoshopped. I really am only 3 feet tall. So Matt helped us pay out the rope. Now, hopefully that'll start up again. Yeah, there we go. When the rope is tight, all 1,200 feet of the rope, remember the other end's tied on the reef, we pay it out, we move backwards away from the reef slope, pay out the rope, and when it's tight or taut, then you drop the trap over. We're over deep water. It'll sink down to the bottom, which will take it down to about 850 feet. And we leave it out there for 2 nights. And then we send Marge back out to untie the rope. First, we have to tie a line from the boat to the yellow rope, the trap line, then untie the trap line. Don't do it the other way around, or the line just sinks down to deep water, and you can't pull it yourself. Then you bring all the line back up, use the windlass, have Marge pull it all up while I continue to take all the pictures. But I don't want you to think she did all the work. I pulled up the last 5 feet of rope, and we had Marge bring the trap in on the boat. That was a great boat to work on for real research. I highly recommend contacting Pat and Lori. There were 16 animals in this particular trap. We have to get them out of the trap. Obviously, they've just come from very cold conditions, and now they're up on the surface. We took tentacle samples from 86 animals. Those samples went to the American Museum for DNA testing. What I've done here, you take the animals, turn them upside down, look inside, and those horseshoe-shaped glands in the back are the nidamental glands, or the shell-forming glands. Obviously, it's a female. Only the female lay eggs, so that's how we sex them. Next thing, dry off the shell, a little alcohol, and write a number on it. We're releasing all of these. We caught them again. We wanted to know that. As it turned out, we didn't have any recaptures. Then Marge took a picture of each animal, profile, and a top view. This happens to be a female, very narrow. Had digital calipers to measure the maximum diameter of each animal. And then back in the cooler. We caught 9.6 animals per trap. And 30 years ago, they were catching 8 animals per trap. So we didn't do very many trap sets, only 10, but enough to get an idea, at least, that the population probably hasn't changed much in 30 years. So that's good news. And not surprising, nobody in Palau catches them. They don't kill them there. It's not like the Philippines. So at least one population is still good. Now, here we are. Marge is releasing the animals at 130 feet. We took them down that deep to kind of give them a head start back to deep water. It's also cooler. But we had something amazing happen I'd never seen, actually horrible. See that Titan triggerfish that just swam by? That fish followed us from the surface as soon as we got in the water, all the way down to, it knew what we had. These fish never ever encountered chambered nautilus in their natural life, but it knew what we had, and it came back for another one. I was horrified, but you can't do much. And I'm a fair distance away. I have the camera on telephoto. But it grabbed this animal, and I photographed it going up. Then I went up after it. So I am going to save that nautilus. It took it back up to 60 feet where Titan triggerfish belong. They don't belong at 130 feet. And he was stuffing him in a little cave up there. Titan triggerfish is the nastiest fish in the ocean. If they have a nest, you don't want to get within 50 feet of them. And it was going to attack me just trying to get his food out of that. It already killed it, though. I couldn't save it, but I did keep the shell. I still wasn't going to let him eat it. But we figured out what was going on. And this was a safe release here, letting this animal go. We protected it all the way down. This spot has a buoy. And we think what happens when the dive shops collect nautilus for photographers to take pictures, they conveniently tie up at this buoy, go down time after time, and release the nautilus for photographers, and then let all the rest go. And the triggerfish have learned what's in those bags. And we think that's what's happened, because we went different areas on either side to release the animals. There were no triggerfish bothering us, and so that's what we did. We're going to send this video back to Palau, to the dive shops, and recommend that they go to other areas to release the animals or follow them down. So anyway, that was a horrifying thing, but it's a lesson well learned, and maybe we can protect more nautilus in Palau at least. Well, I'm going to end on this slide. I looked at this video when I was putting my presentation together, and it reminded me what I said earlier, that our exhibits should exceed visitors' expectations. And I think that very few visitors expect to go diving in an aquarium. And they should try to create an emotional connection with the animals we display. And I'm sure that's what's happening here, maybe for the humphead wrasse as well as the divers, two-way mutual emotional connection. And I certainly hope our aquariums, whether they're home aquariums or public aquariums, will help create a greater understanding of ocean life and stimulate a commitment to conservation. And I think aquariums, again, hobbyists or public aquariums, really have tremendous value in that regard, in the ability to stimulate those kinds of interests, and particularly in kids. I hope that aquariums will continue to inspire young people to pursue their own careers in marine biology, and that's how I got my start. It's been a very exciting 50 years for me, and thank you very much. Okay.